Jacob and Monod first gave us the operon concept or the model of the operon. Here we will restrict our understanding only to the prokaryotic system. All right. Now, and it has been studied in, so their study is based on the operon system which is found in the bacteria Escherichia coli. Okay. There are different components of the operon system. So there will be a structural gene. Then there will be an operator gene. There will be a promoter gene. And lastly, there will be a regulator gene. So these are the different components that are present in an operator system. The operator system that we will be studying is lac operon, and that is a an inducible operant system. Inducible meaning that the synthesis of lactose is not always taking place. It is only taking place when the body is requiring it. So here I take a segment of the DNA. This is a segment of the DNA, and this segment of the DNA is having several genes on it. What are the genes? First, we have an area. Suppose this area on the DNA is the area for the structural genes. This is for the structural genes. Just ahead of this, now if this is a DNA, then this will be the 5' end and this will be the 3' end. All right. In the DNA, the 5' end and the 3' end are present in that manner. Okay. Towards the 5' end, the regulatory sequences will be present. Towards the 5' end, the regulatory sequences will be present. That means just ahead of the structural gene, there will be the presence of the operator gene. This is the site for the operator gene. The operator genes will be present here. Beyond that, this site will be for the promoter gene. Operator gene is denoted by O, promoter gene by P. Then the site here, this region is termed as the regulator gene, which is shown as I, and this region is known as the promoter site or promoter site for the regulator gene. Promoter site for the regulator gene. So it is termed as PI. On the PI site, the RNA polymerase will come and rest. This is the RNA polymerase for the regulator gene PI. Alright. Now, if the RNA polymerase comes and sits here, then it will initiate transcription. When transcription is initiated, then this regulator gene here, the regulator gene here will synthesize a protein it synthesizes a protein. All right. Now, see, suppose this is the protein. This is the protein. All right. Now, there is another promoter site. So, at this promoter site, another RNA polymerase will come and sit. All right. Another RNA polymerase will come and sit, and this RNA polymerase is for the synthesis of the structural genes. All right. Now, this promoter gene will thus be helping in the synthesis of another protein. All right. So, what will happen? This protein will be taken up by this protein in this manner. Okay, right? So, what is happening? This protein is unable to go and sit at the operator site. It is unable to move to the operator site. Its pathway towards the operator site is blocked. If the pathway to the operator site is blocked, then the RNA polymerase can progress its transcription process and the structural genes will synthesize the required polypeptides. Is which is known as the inducible operon system. Why it is inducible? Because it is allowing the synthesis of the structural genes. Lac operon comes from the word lactose. So the regulator protein here is lactose. All right. Breakdown of lactose occurs with the help of three enzymes. What are the three enzymes? Lactose breakdown occurs with the help of three enzymes. What are the three enzymes? The three enzymes are beta galactosidase, permease, and trans acetylase. Now, each of these enzymes, that means they are proteins, will be regulated by three types of structural genes. What are they? The structural gene for beta galactosidase is known as gene Z. For permease, it is known as gene Y. And for trans acetylase, it is known as gene A. Okay, you'll have to remember these things. So, what is the function of each of these enzymes? What is the function of each of these enzymes? Now, let us look at that. The gene Z, because it is associated with the lac operon system, so it is known as lac Z. It is known as lac Z. Similarly, here it will be known as lac Y, and here it will be known as lac A. Now, what is the function of this? This is this is coding for beta galactosidase. And what does beta galactosidase do? It hydrolyzes lactose to glucose and galactose. Why we call hydrolysis? Because the water is removed, water is added. Water is added to the glycosidic linkage and the cleavage occurs. How is the linkage created? How is the linkage created? By the bond between two consecutive monosaccharides. All right, and when so the lactose molecule is having a glucose and a galactose molecule forming a linkage, and by hydrolysis this glycosidic linkage is broken to result in the monomeric units, that is glucose and galactose. What is the function of LAC-Y? LAC-Y helps in the synthesis of permease, or truly speaking, we can also call it as uh, beta galactoside permease, to be more specific. So, what does this do? It increases permeability of membrane. What membrane? The plasma membrane. It increases the permeability of membrane to lactose. What membrane? Plasma membrane. So, what does this indicate? That always there should be some amount of permease present in the system to allow lactose into the cell. Otherwise, this operon system will not be functional. It cannot be made functional. Lac A codes for trans-acetylase. So what does it do? It transfers the acetyl group. It transfers the acetyl group from acetyl coenzyme A to beta galactoside. It transfers the acetyl group from acetyl coenzyme A to beta galactoside. What are the two binding sites? One is for the operator. When can it go and bind to the operator? When the regulator protein did not come and attach to it. And the other is for the lactose. The other is for the lactose. All right, and it is always functional. It is that if the repressor protein binds at the operator gene, then switch off mechanism occurs. That is, RNA polymerase progress is hindered. In which direction RNA polymerase progress will occur? Towards the structural genes. Alright. But if we have the repressor protein binding to the inducer. Which will act as the inducer here? Lactose. If it is binding to the inducer, then a switch on mechanism occurs, like the one that I have drawn. All right. So RNA polymerase progresses towards the structural genes.